Tissue regeneration and regenerative therapies are the future of medicine.
If we can recreate the lung outside the body, we can use these for transplantation approaches.
We consider it one of the most fascinating biological questions, the ability of organism to completely repair a lost appendage, a lost limb. It's truly remarkable.
Our overarching goal is to make this a center for tissue regeneration, to have people come here to study it, incorporate it into their innovations, into their practices, to grow and to make a difference. We've hired experts in gene therapy of regeneration, in how the lung repairs itself after injury and disease, genetics and genomics of tissue regeneration, and stem cell biology.
Where heart regeneration is really exciting is that it could actually cure the actual problem that's causing the disease, replace the lost or dysfunctional heart tissue. My lab is really focused on 3 Ds: detection, discovery, and delivery. Identify factors that can actually drive heart growth. And then after we know what those are, then we actually have to figure out how to get it to the heart. After that, you have to know if you made people get better, and so you have to be able to detect generation.
Kidneys are one of the least regenerative organs in the body. About 50% of patients on dialysis would not be living in 5 years. I'm hoping that we could develop models or systems that could replace lost function of the kidney by understanding how the tissues develop naturally and using that information to guide how we design our differentiation strategies. We're able to generate tissues and cells that are much more specialized. And I think effectively developing models that could better recapitulate organ level functions and the mechanisms involved in them as well.
We've been building our faculty. We've been building our ability to recruit trainees, students, and postdocs, and we've been giving them support, grants, and fellowships.
The big question our lab studies is how complex organs form from simple tissues in a very robust and reproducible manner. We can use our genetic and genomic studies to address human diseases. 70% of human genes have an ortholog gene in zebrafish. They're transparent, so we can watch their development live at high spatial and temporal resolution.
How can that tissue remember how big it was or grow back to the same shapes, and how can you do that reproducibly every time? That information must be encoded somehow. If we could start understanding how any of this system encodes that memory, that would really bring a new way to think about regeneration.
We've brought together scientists and clinicians and engineers to meet and talk about regeneration in large community settings.
Regenerative medicine can make major improvements in the outcomes for our patients. One approach is to be able to, in a lab, take cells and make them grow into new cartilage and put them back into people. Another way is to inject cells into the joint that might regrow and make new cartilage inside the body. A third way is to be able to give drugs or other kind of therapies to make your own cells regrow those joints.
In my lab, we hope to understand the mechanism that control the cell state change of muscle stem cell in regeneration. We are also interested in how niche population communicate with muscle stem cell. And also we hope to extend our analysis to some disease models such as muscular dystrophy and aging. Duke Regeneration Center provide an excellent environment for us to carry out our research to address all these questions. So we are able to collaborate with developmental biologists, bioengineers, physicians, and surgeons to address the fundamental question of muscle regeneration.
Duke has one of the largest lung transplantation program, so we are side by side comparing these so that we can relate anything that we learned in the model systems in human. We identified new cell types in human lungs that we never knew before. Being able to modulate and manipulate these cell types may offer avenues for regeneration.